Hi, welcome to this episode of CPN TV. I'm Alison Robertson. I'm a professor and extension plant pathologist at Iowa State University. And today I have a couple of colleagues that I've been working with. Asela Wijeratne, who's an associate professor at Arkansas State University, and Brett Hale, who just finished his PhD with Asela at Arkansas State University, and Brett works for AgriGrow. And today we're going to be talking about some of Brett's research that he did. And the title of this episode is Stopping a Soybean Killer, a Systems Biology Approach. So what is the soybean killer that we're going to be talking about today? Phytophthora root and stem rot. So this is a disease that of soybeans that is probably familiar to a lot of you. And I'm just going to go over a little bit about the disease, just to remind everyone about this disease. And then we'll get into some of the research that Asela's doing and some of the research that Brett did for his PhD. So Phytophthora root and stem rot is a disease of soybeans. It causes losses every year in soybeans. And the one thing about this pathogen or this disease is that it can occur at any growth stage. So we can see damping off of seedlings in the field. We can see young plants that are dying soon after emergence. And then we can see older plants also that die. The one thing about Phytophthora is that when we look into fields, often we see large patches that occur in fields, but we can also find individual plants that die. So when we look at those Phytophthora root and stem rot plants, the symptoms that we're looking for are, first of all, as you peel back that soybean canopy, what you're going to notice is that the plants are yellowed and wilted. And when we look at those plants, you're going to notice a dark chocolate brown lesion that kind of extends from the taproot up to the stem. Another thing about these plants is they're very easy to pull out of the ground because there's a lot of root rot that goes on. So as I mentioned, Phytophthora root rot, I love this picture. I think it's a great picture just showing that chocolate brown lesion coming all the way up from the taproot. And it's quite easy to get Phytophthora root rot compared with or confused with another disease, and that would be stem canker. And what I want you to notice here about stem canker is that we also get this brown lesion on the outside of the stem. But because stem canker starts, the infection starts at the nodes of the soybean, that means that below those nodes of infection, so at that soil line, you should see green tissue. So Phytophthora root and stem rot and stem canker are the only diseases of soybean that will cause this chocolate brown lesion on the outside of the stem. Last year, in 2023, we had a big outbreak of Phytophthora root and stem rot across the Midwest. And these are some tweets that were sent out by a lot of soybean pathologists that are working on soybeans. And so the amount of Phytophthora root rot that we see will kind of wax and wane from year to year. And for those of us working in Phytophthora root and stem rot, 2023 was a particularly good year for the disease. And a lot of us got very excited to see these symptoms out in the field. So to manage Phytophthora root rot or to reduce losses from Phytophthora root and stem rot, we need to manage it. And the best way to manage this disease is to take an integrated approach. So we can grow resistant varieties. There are also seed treatments that can be used. We can also improve drainage. And I'll get to in a moment why that's important. And then lastly, if you think about integrated pest management, a lot of times we'll recommend things like rotation and crop residue management. But for Phytophthora root and stem rot, that doesn't really work because of the biology of the pathogen. So the best way to manage Phytophthora root and stem rot is using resistant varieties. The problem with the resistant varieties is that they are not resistant forever. Eventually, that pathogen evolves and overcomes that resistance. And so that's kind of that's the inspiration behind the work that goes on in Asela's lab. So I mentioned Phytophthora root rot. So what is it caused by? It's caused by this pathogen called Phytophthora soji. And the most important thing about this pathogen is that it survives in the soil as oospores. So you can see this a picture of an oospore there, and you can see this very thick wall around the oospore. So that enables the pathogen to survive for many years in the field. And that's why rotation doesn't work to non-host crops, because that oomycete will just survive for several years in the absence of soybeans. When those oospores germinate, they produce these lemon-shaped sporangia, and within those sporangia are zoospores. And the zoospores are what infect the soybean. And the thing about these zoospores is they have flagella on them, and so that enables them to swim in free water. So that's why improving drainage in a field can help manage Phytophthora, because by improving that drainage, you're not getting these zoospores that are swimming through the water to find the soybean roots. We've already talked about the symptoms of Phytophthora root rot, but if we take some of those infected tissues and put them on a Petri dish with a special kind of jelly, what will happen is the Phytophthora soji will grow out of that jelly. And so you can see it kind of looks like a fungus growing out of that mycelium, that fluffy mycelium that you see is the Phytophthora soji. Okay, so now I mentioned that in Asela's lab, they work a lot on resistance. And so I'm going to pass the mic over to Asela, and he's going to tell us about how resistance works in this pathosystem.
Yeah, thank you, Alison. So what we have here is what we call a cross-section, a cartoon of a cross-section of a root on your right-hand side. So as Alison mentioned, some plants are resistant to this pathogen. So how does that happen? So when the plants are resistant to the pathogen, they carry a gene called RPS genes. They actually make a protein that can detect certain pathogen secreted proteins. When that happens, that plant protein can then activate a set of downstream genes, or a set of genes that can then go on and activate defense mechanism, which eventually kills the pathogen. So that's how the resistance work. We can talk how this actually, how pathogen can evade this in the next slide. Okay, so here's again, very similar cartoon. So as Alison mentioned earlier, some plants loses this resistance. How does that happen? So if you keep growing resistant plants in the field, what happens is that the pathogen can continuously see the resistant plants. So what they're going to do is they're going to change some of their molecules, in this case, what we call the effector proteins. They're going to change the structure of those proteins. And then what happened with that is that the plants will not be able to detect these change or modified defectors. So with that, the pathogen can bypass the surveillance mechanism and then basically can invade the plant. And here's has a cartoon showing eventually if the pathogen can bypass the surveillance, that it can go on and invade the root, and eventually, as we have seen in the previous slides, it can eventually kill the plant.
Thanks, Asela. If you're interested in this gene-for-gene interaction that occurs in Phytophthora, and then how we as pathologists use it to characterize the Phytophthora soji in fields, there's another CPN TV episode by Marty Chilvers Austin McCoy, taking the headache out of managing Phytophthora in soybean. And they do a really nice presentation about this gene-for-gene interaction and how we can characterize Phytophthora soji and understand more about the pathogen in the field and therefore manage it with resistant varieties. So, Asela, do you want to explain what you're doing in your lab or the goals of your lab?
Yeah, so what we are mainly interested in the pathogen plant and the environmental, all three component interaction. So these three components, we typically in plant pathology, we call it disease triangle. So eventually, if you see a disease, these three components needs to interact in a certain way to see a disease outcome. Traditionally, most plant pathology research has been focused on the plant pathogen host interactions, but we want to also focus on the other parts, which is the environment. In this particular case, we have seen the microbiome, like our gut, the plants also do have microbiome in their roots. They influence a lot for this outcome. So I'm interested in my lab, we're interested in looking at these microbes as well to see how they impact the disease outcome. The other part, which we are going to discuss in detail today as part of Brett's work, is to look at what happened after the initial pathogen recognition. What happened in the cell after this initial recognition? What are the other components in this process that we can use eventually make these soybean plants more resilient against the pathogen? So those are the main two components of my research. Yeah, so this kind of goes back to what I mentioned earlier. So we are, one of the main interests in my lab is to look at how these plants, when plants recognize, or in this particular case, soybean plants, when they recognize the pathogen, what happened downstream. So there are, based on previous research, we know that there are two pathways involved in this process. Basically, one pathway is called PAM-triggered immunity, that the idea is that this is like basal immunity. So in other words, even in human, we do have immunity to all the pathogens or all the microbes that we get into contact with. Similarly, there's PTI, or the PAM-triggered immunity, is more general immune system or a way to suppress the pathogen. The second category that is involved in this process is we call the effector-triggered immunity. The idea here is that every pathogen has a specific recognition mechanism through this pathway, and then that can activate a very robust immune response against the pathogen. So PTI is more like a general surveillance mechanism. ETI is a very specific surveillance mechanism that can activate a robust immune response against the pathogen. So those are the two pathways that we are looking at. So these are kind of molecular mechanisms that we are looking at.
So the question then is, we have these plant pathogen interactions. Soybean can recognize PSOJ. It's eliciting a robust immune response whenever it has the correct RPS genes. It's only able to elicit the weak immune response, which is suppressed whenever PSOJ gains the upper hand. Nevertheless, what's happening with the rest of the genes in soybean upon PSOJ infection? How are they behaving? And if we have a better understanding of how they are behaving and how they are controlled, that opens the door for new avenues to potentially breed soybean to be more resilient to PSOJ in the field. And there have been several studies that have looked into this. For example, it's been shown numerous times that upon PSOJ infection, thousands of genes start behaving differently in soybean. A study in 2014 showed that there were 5,500 genes that changed. Other studies with slightly different approaches have shown upwards of 25,000 genes that were behaving differently. So we have this global reprogramming essentially at the molecular level in soybean in response to PSOJ. And then that begs the question is if we have these global changes, if all this is happening, again, what are the controllers of these changes? And can we get that information and leverage it to our advantage to help control the disease?
Great. So what did you find, Brett, in your research?
Well, the first thing that we wanted to do was see if we had similar results, if we could get similar results to what's been reported in the literature. So we had two different interactions that we set up. So the first that we did is we took soybean and we inoculated that hypocaudal region with a kind of PSOJ that it could recognize and then elicit that ETI, that robust immune response. And if you look on the bottom left, that's where you're seeing the no disease, the PTI plus ETI. You see that brown lesion, but essentially you have that localized cell death, and that's a resistant interaction. When you look on the right, this was another treatment we used where we essentially inoculated the soybean with a kind of PSOJ that it couldn't recognize that is suppressing that basal immune response. And you can see where that hypocaudal tissue is soft, it's rotting, and you have that expansive lesioning. We had a third treatment where essentially we just inoculated those soybeans with that sterile jelly we talked about earlier. That's not going to, it's not going to have the pathogen. We're not going to have that disease pressure. And then we compared how the genes were behaving in this resistant interaction compared to the sterile mock, as we're going to call it, and also in the context where we had the disease compared to the mock. And what we saw was we had over 6,000 genes that were behaving differently in total between these two infection interactions compared to that mock, which is consistent with the previous literature. And you can see at the time point that we selected, many of them were specific to this disease interaction, which you can see on the right here in this Venn diagram in the yellow. Another sliver, a small sliver of 175 were specific to this interaction on the left where we have resistance. And then we had a big chunk in the middle where the genes were behaving differently in both infections compared to the mock. And again, this is just illustrating the point that we mentioned earlier, what the immune response is looking like in each of these contexts. So if you look on the left, this is what we're seeing where we're not getting disease is we have this general recognition of the microbe, kind of hard to see up here at the top. This recognition is leading to PTI, which does give us transcriptional reprogramming. Whenever the pathogen then secretes the effectors, we have this protein. This here is essentially it's representing the RPS protein. We're recognizing the effectors, and we're getting this robust immune response, which again leads to transcriptional reprogramming. And then in the context here on the right, of course, we're only getting the PTI response, and then these effectors are suppressing it. We're not getting the ETI. Okay, so we had, like I said, 6,000 genes roughly that were behaving differently when PSOJ was infecting the soybean tissue. So the next thing that we did is we essentially clustered these genes based on their behavior. Are they being expressed at a higher level where the PSOJ was added, or are they being expressed at a lower level? And when we did this clustering, we could essentially cluster these 6,000 genes into 9 clusters. And that's what's being represented here on the left with these red and blue blocks. So there's 2 columns, and the one on the left, again, is this resistant interaction where we don't have disease. The column beside it to the right is the interaction where we have the disease. And what's really interesting is you can just generally look at the color and see that all but 2 of these blocks are red. These are genes that are being upregulated. They're more active. And we have 2 blocks with genes that are doing the opposite. They're expressed at a lower level. They're not as active. So that was really interesting. So the next thing we want to know is, what are these genes most likely doing? We have these really interesting clusters. What is this behavior signifying? And we have these bar plots next to it. And I'm not going to get into what's being said in each of these blocks because it's summarized on the right. All of the clusters of genes that are red, that are being upregulated, that are being more active, are related to the host immune response in some fashion. They're important for fighting off PSOJ, whereas the other 2 that are being downregulated, the genes are becoming less active. Those are clusters of genes that are important for general growth and reproduction. So it's really interesting because it looks like the soybean is taking its energy and its resources and putting it into defense rather than photosynthesis and general metabolism.
That's really cool, Brett. But how do we turn genes? I mean, and it sounds like the soybean plant is turning genes on, and then it's also turning genes off. So how does that happen?
That's a very good question. And there are several mechanisms behind gene regulation that warrant their own discussion. But one common way is through transcription factors, which are proteins that act as these master regulators to control gene expression. So based off of these trends in gene behavior, it appears that the soybean plant is balancing primary growth and reproduction and the immune response. So it's essentially, when infected with PSOJ, it's taking its resources and it's allocating it toward resistance or toward an immune response and away from photosynthesis and this primary growth. So this is not uncommon. This has been seen in other systems as well between plants and their pathogens. But it was really interesting. We saw it very clean cut in this study.
Brett, that's really interesting how those soybean plants can either turn genes on or turn genes off. So how do they manage to do that?
Yeah, that's a very good question, Allison. And there are a lot of components of gene regulation, which get very complicated. One component of that involves transcription factors, proteins that actually can act as on and off switches for genes. And with that, I'm going to turn it over to Asela to discuss the central dogma molecular biology and give us a little more background in transcription factors.
Yeah, so the idea of central dogma of molecular biology is that every cell, I mean, every living organism has this blueprint of DNA, right? So, but not everything in this DNA has instruction for the function. So there are certain entities called genes that are important for eventual function of this functional information. So what happened is that in a cell, these genes can make what we call an informational molecule or messenger molecule called RNA. And that RNA will go on into what we call the cytosol of the cell. And then it can instruct the cells to make proteins. This process from going from DNA to RNA to protein is what we call the central dogma of molecular biology. Also, what is important here is that the workhorses of cells are proteins. So that's what actually do most of the function in a cell. So transcription factors are one type of proteins. And as Brett said, they can turn off and turn on the transcription or the making of RNA in a cell. Yeah, so this cartoon shows exactly how they work. So these transcription factors, as I said, they are type of proteins. They can bind to a certain region of the DNA. We call those promoters in most cases. And they can then increase the production of RNA, or it can decrease the production of RNA. So if it is increased, we call it upregulation. If it is decreased, it's called downregulation. So this process is transcription factors can control this process. Transcription factors are not the only molecules, but they do a huge role in this process. Okay, Brett, I will turn over to you, and you can discuss what are what some of your findings in regards to transcription factors.
Sure. So we're going to work through this. Looking back on the left, we see these columns that we saw previously of these genes that are clustered. We have the red, we have the blue. We know that the genes that are in the red clusters correspond to those important for immunity, and those in the blue clusters are important for growth and reproduction. So knowing that we have this global reprogramming of gene behavior, and knowing that this process is at least in part mediated by transcription factors, the next question is what transcription factors are behaving and how to mediate this response. So what we did is we looked to see genes that actually make these transcription factor proteins, and look to see, out of all these genes that are behaving differently, which genes are making transcription factors, and then within each of these clusters, how many transcription factor encoding genes we can find. So we did that, and that's what's being depicted here in the green, showing transcription factor families. So transcription factors are categorized into families based off of their structure and their function. So we're looking at the families of transcription factors and their representation essentially in each of these clusters. And that's what we have here in the green. Okay, when we move to the right in this plot, the next thing we did was to see if any of these transcription factor families, these families of proteins, if they are represented more or less or enriched for in this set of genes that are behaving differently, okay? So we use two different metrics to look at that. We look to see how many genes that make these proteins were present in this set of 6,000. We also looked at the percentage of the family. So for example, maybe you have a transcription factor family. There's only 5 of these proteins in soybean, but all 5 of them seem to be behaving differently because of the PSOJ, because we're adding the pathogen. That's a family that is significant and warrants further investigation. So we use these two metrics, and we looked here, and what you can see is that the Werke transcription factor family and this ERF family were the most abundant by the total count. They really stood out to us. There are some other families that are labeled here that are known to be important in plants for controlling a stress response. And then we had another family here on the far right that there aren't many of them, but again, nearly all of them, the genes that make them are behaving differently, called RAV. That was another family that warranted further investigation. So as we continued to compile this story and learn, we really focused on these families, Werke, ERF, and RAV. And this warrants the question, again, what transcription factor target gene interactions are fine-tuning the defense growth trade-off in soybean following PSOJ infection? So we know that transcription factors are important in this process. We know we have these genes that are behaving differently. We have candidate families of these transcription factors. How does all of this work? And to help answer that question, we'll turn it over to Asela.
Yeah, so transcription factors and how they regulate the gene expression, or as I said, how they increase the production of RNA or decrease the production of RNA. It's actually kind of really complicated process. They sometimes do interact with their targets directly. They go and bind to the targets, or they can do it via another transcription factors. And there can be multiples of these. So what we see is that sometimes they form this what we call a network. It's like a city traffic, right? So it can have different roads, different roadblocks, can have signal lights and all that, and then it's very similar to that. And then some of these components of this network can be more important than the others. So what we are trying to do here is to build this network. We call it gene regulatory network. And once we build this network, we can identify, okay, these are the most important component of this network. And then we can start investigating those most important components. Okay, so in the lab, we use a special technique to find these transcription factors and their interacting partner. And that's one step to build these networks that I mentioned in the previous slide. So using this method, we could identify if you have a transcription factor candidate, we can use that, and then we can identify which target genes that it might be regulating. So I'll turn over to Brad to discuss some of his interesting findings.
Okay, so we zeroed in on transcription factors that belong to two of the families of interest. So we focused on one that belongs to Werke, and we focused on another that belongs to RAV. And the genes that make both of these transcription factors were behaving differently in our study. So these were really good candidates to work with. So based off of that binding site assay that Asela told us about, we found targets for this Werke transcription factor, 212 putative target genes that were behaving differently. And when you look to see the functions of those genes or what we think they're doing, they mostly correspond to genes important for the immune response. So it seems like this Werke transcription factor is really important in regulating that immune response in soybean. Contrastingly, the RAV transcription factor that we looked at, the potential targets of that included 389 genes that were behaving differently. And when you look at their functions, it included genes important for primary growth, reproduction, and the downregulated genes. So it's interesting. We found 6,000 genes that are behaving differently because of PSOJ. They clustered into these really neat clusters, some important for immunity, some important for defense, and they're showing opposite trends in behavior. And we've identified now potential regulators of both of those processes. And related to that, the Werke 30, this is the transcription factor that we did the assay with. We just showed you the gene that encodes that, the gene that makes that protein. This is how it was behaving in the original study when we were looking at the gene activity. And you can see that it's expressed at a much higher level where disease occurred versus where we had resistance, which was really interesting, knowing that the protein that comes from this, the transcription factor, presumably increases expression of the immune response genes. And this is really interesting because this could be just a response related to a heightened immune response. This could also be one of the mechanisms of the pathogen to misregulate the activity of this gene to help hijack the defense signaling pathway. This is all very coordinated, and we want certain levels of these things to be behaving in a plant cell. And if you get misregulation, that could be problematic for the plant.
Those are really exciting findings, Brett.
All right, and I can tackle the conclusions then. So ETI and PTI, these two components of the plant immune system, they serve as plant bodyguards. And then ETI works to boost PTI's work. As we said, most of the time PTI is sufficient. It's good enough for non-host resistance. However, when you have a pesky pathogen that's lingering on a particular host plant, like PSOJ does on soybean, you need more. You need reinforcement, and that's where ETI comes to work. And threats trigger a lockdown in plants, which we call transcriptional reprogramming. That gets back to the gene behavior and having this global response where these genes start working differently. And this is characterized by defense genes being activated, which is like calling reinforcements, and then normal growth, reproduction, metabolism slows down. It's like closing the non-essential services, or at least they're not essential in that particular moment of crisis. And certain proteins, like the transcription factors we've been discussing, they fine-tune this process, and they act as the commanders.
Thanks, Brett. So Asela, Brett's left the lab now and gone on to greener pastures, perhaps. So what are you planning to do now?
Yeah, so these are some of the observations that we have made. So as scientists, we need more definitive answers. So we always try to find more information to support our hypothesis. One of the things we are doing right now is taking these genes and look at their function more closely. One of the cooler tools that we have right now is, you probably have heard of this, is the genome editing technique called CRISPR-Cas that allows us to remove certain part of a gene or insert certain part of a part into a DNA. We can do a lot of different things using this genome editing technology. What we are doing with this genome editing technique in my lab is to remove the function of these genes. So basically what we are doing is that we mutate these genes so that they are no longer functional in resistant plants. So initially those plants were resistant to PSOJ, and then now we mutate these genes, and then we infect those mutated plants with the pathogen. So if those genes are important for defense response, as we discussed, those mutant plants will become susceptible. So that's the idea, and that's what we are trying to do in the lab, and try to gain more understanding of the functions of these genes.
That's really cool, Asela. Can you explain to folks who might be more applied and in the field, how is all this information going to be important as we move forward with managing Phytophthora root rot on soybeans?
Of course. Yeah, so there are different ways we can improve crop productivity and making them more resilient against pathogens. We have conventional breeding, we have mutation breeding, transgenic breeding, and then there are this novel technology, as I mentioned in previous slide. You could also use gene editing and gene silencing technologies to change the behaviors of certain genes. And we can use what we find in our lab in any of these methods. For example, as I said, the defense response is regulated by many different genes, right? So we can, once we identify those genes, we can now target those genes using one of these techniques and say, okay, now we want more of this gene product, or we want less of this gene product. We can manipulate it in a way that makes these plants more resilient, and also that resistance can be last longer than what we are seeing right now with the RPS genes. So that's the idea, and that's what we are trying to achieve at the end.
That will be great, because we certainly need plants that are more resilient to this pathogen that just manages to overcome that resistance time and time again.
Yeah.
If you're interested in the gene editing that Asela's been talking about, Don Lee from the University of Nebraska-Lincoln also did a CPN-TB episode where he discusses RNAi, and he goes into a little bit more detail about how we can edit genes using those tools.
Yeah, so most of this work was done by Hale, as you have already heard, and then there are other people in the lab. Ashley Flory did some of the DNA protein binding assays, and then Sandra Ratnaker. We didn't get to talk about his work, but we expanded this work to many other transcription factors using machine learning. And Sharon Portal is also helped with some of this work, and in Alison's lab, Clarice Schmidt. Without her, we won't be able to do any of this. She did all the plant pathology work. And also want to acknowledge some of our funders. We got money from College of Science and Math at A-State, and then Arkansas Bioscience Institute. And we also got some funding from INBRE. And also I want to acknowledge, High Performance Computing Center for all the support they have given us for data analysis.
Well, thank you, Asela and Brett. Thanks for joining me today. And for those of you who are watching this episode, I hope that you've enjoyed it and learned a little bit more about how we can use our understanding of how genes interact between a plant and a pathogen to hopefully develop more resilient plants to manage those diseases and protect those yields. So thanks for joining me today.